going to talk about the neuroscience of gender identity. I'm going to specify more in the neuroanatomy in humans that identify as transsexuals and structural brain changes after hormone replacement therapy. So our first paper is called A Sex Difference in the Human Brain and its Relation to Transsexuality. A little information about the author. His name is Dick Swab. He's a professor of neurobiology at the University of Amsterdam, which it was here he got his PhD. He is mainly recognized for his work on the relationship between sexual dimorphism and brain anatomy and research on transsexuality and sexual orientation. Some general background, I think it's very important to know the difference between the sexual orientation and sexual identity between, a person. So sexual identity is your sense of self in a relation to your sexuality. As such, it encompasses many components, including gender identity and sexual orientation. Sexual orientation is the term used in regard to who you're attracted to or sexually and romantically. Another main important brain area that is discussed in this paper is called the BST, also known as the bed of the nucleus of the stria terminalis. And it's a brain area that is essential for sexual behavior. Some methods they conducted in the experiment was that they searched for a brain structure that was sexually dimorphic, but that was not influenced by sexual orientation, as male to female transsexuals may be orientated to either sex with respect to sexual behavior. For a variety of reasons, the bed of the nucleus of the stria terminalis was proved to be a good candidate for this investigation. First of all, it was well recognized that the BST is crucial to rodent sexual behavior. And some other animal experiments concluded that the BST is the key for aromatization in the development of rat brain and has both oestrogen and androgen receptors. Also, in humans, a particular caudal part of the BST has been reported to be 2.5 times larger in men than in women. Regarding the results, in comparison to the heterosexual women, heterosexual men had a 44% greater BSTC volume. Men who identify as heterosexual and homosexual did not have any statistically significant differences in the BSTC volume. Compared to straight women, the BST in homosexual males was 62% bigger compared to women. And a small volume of the BSTC was found in male to female transsexual. Its size was only 52% of that found in the reference males of 46% of the BST of the homosexual males. As all the transsexuals have been treated with oestrogens, the reduced size of the BSTC could possibly have been due to the presence of high levels of estrogen in the blood. Another cool fact about this experiment, it was that this occurred in the AIDS epidemic. And so they did research on how the size of the BST was influenced by AIDS. So they found that AIDS did not seem to influence the size of the BSTC. The size of the BSTC of 2 heterosexual AIDS-infected women and 3 heterosexual AIDS-infected men remain well within the range of corresponding the reference group. In the overview, the observations suggested that the small size of the BSTC in male to female transsexuals cannot be explained by differences in adult sex hormone levels, but it established during the development by an organization in actions of sex hormones. There was no correlation between BSTC size and sexual orientation of transsexuals, whether they were male or female orientated or both. Furthermore, there was no difference in BST size between heterosexual and homosexual men, supporting the notion that the smaller the BSTC size is unrelated to sexual orientation. Regarding the second paper, we have Mark Breedlove, which is a professor of neuroscience at Michigan State University. Breedlove had demonstrated that steroid hormones and sexual behavior affect the development the developing an adult spinal cord and brain. He also have reported in several studies that the average digit ratio of lesbians is more masculine than that of straight women. In his paper, another important organ, we have, he stated that post-mortem analysis revealed the volume on his brain region is smaller than in men, and that 6 transsexual men also had small BSTC. Thus, men who wish to become women had a small BSTC than other men. And it also, he stated that the BST size is not related to sexual orientation. Regarding the paper, he also reported a sex difference in the human preoptic area 7, which is a region they name the sexually dimorphic nucleus of the preoptic area, also known as SDN-POA, after all the similar nucleus was found in rat brains. The SDN-POA was larger in adult men than in adult women, but this group reported that there was no sex difference in the number of neurons in the human SDN-POA until the 10 years of age. This leads to the fact that there is a ample room for early experiences, including the very different social stimuli presented to boys and girls, to the cause of sex difference found in the adult SN-POA. Lastly, the third paper is called Changing Your Sex Changes in Your Brain, Influences of Testosterone and Estrogen on the Adult Human Brain Structure, which is reported by Haley K. Holshow-Pohl, she's a psychologist and researcher in the field of neuroscience. Her research focuses on which genetic and environmental factors influence changes in the brain and how these influences are related to cognitive functioning. She studies the changes in brain structures and functionings during prenatal and postnatal development aging. A background on regarding this paper is important to know that transsexualism is the condition in which a person apparently normal somatic sexual differentiation of one's sex is convinced that he or she is actually a member of the opposite sex. This sense is so pronounced and persistent that transsexuals seek treatment as far medically possible to physically change their bodies from a male to a female or vice versa. It is known that sex hormones also promote the organization development of the sexually dimorphic brain. So the objective of this paper is to see how transsexuals who receive cross-sex hormones have unique opportunity to research how sex steroids affect early adult brain morphology. The methods that were used for this paper was magnetic resonance. Magnetic resonance brain images were made prior to and during cross-sex hormone treatment to study the influence of anti-androgen estrogen treatment on the brain morphology in 8 young adult male to female transsexual human subjects and of androgen treatment in 6 female to male transsexuals. The results, according to the research, the anti-androgen plus estrogen treatment decreased brain volumes to male to female subjects toward female proportions, while the androgen treatment in the female to male subjects increased total brain and hypothalamus volume towards the male proportions. Another interesting finding is that a decrease in sexual arousal, sexual desire, and spatial ability, typically in men who outperform females, as well as an increase in verbal fluency were seen after 3 months of estrogen in addition and testosterone suppression in male to female transsexuals. 3 months of testosterone therapy in female to male was linked to an increase in sexual arousal, aggressiveness, propensity, and spatial ability, but not fluency test. According to the research concluding this, androgen therapy raises the female brain's volume toward that of the male, and anti-androgen estrogen therapy decreases the male brain's volume towards that of a female in young adult humans. The results suggest that the mature human brain structure can change to accommodate the other sex under the influence of transgender hormones. Yes.
Okay, so next we'll be looking at a paper from the Arnold lab about a gynandromorphic finch. And before I get into what that means, I'll tell you a little bit about the author. So Arthur Arnold was actually a postdoc at the Breedlove Lab, which you guys just heard about from Diego. He's now a professor and researcher at UCLA, and he focuses on sex differences in the brain due to gonadal or genetic differences. And he focuses a lot on the differentiation between male and female tissues. So before this paper came out, the current dogma about brain sex differences were believed to result from just what the hormone environment of the brain was. So if you had, a certain reproductive organ, your brain would be subject to those hormones within the hormones produced by the reproductive organ, and any cell outside of those organs were neutral and just subject to the steroids that they produced. However, some studies had been incapable of fully sex reversing, for example, female finches to male by providing them the male hormonal environment in the brain, which means that there might be problems in the model because hormones are not the entire cause of sex differences in the brain. And that's when this paper came out saying that there was a neural component to sex differences, not just gonadal. So a gynandromorph is an organism with both female and male characteristics, often divided directionally and on a genetic basis. And the zebra finch is a useful model organism for sex differences in the brain because males are the only ones who sing, and they sing to female for courtship. So that means males have a large song nuclei, including area X, RA, and HVC, which you guys will hear about in this study. And for anyone who's curious as to how they made this gynandromorph, they just found it in the wild and then published a PNAS paper from this organism, which is one of the happy science accidents. So this paper found this finch because the plumage was split down the middle. So on one side, it had the pretty stereotypical like dark gray plumage from a male, and on the left side, light gray from a female. They also noticed that the gonads were split, as you can see on the right figure here, but they also had abnormal phenotypes. So they didn't function quite well, and they actually didn't detect any steroids above the detectable limit, which is important because that means when they studied differences in the brain of this organism, it couldn't be traced to the hormones produced by the reproductive organs because these hormones were at such a low level of production that they didn't really have any effect on the brain. So now looking at the brain in the figure in the middle, they looked at sex-linked mRNA production. So as you can see on the bottom left here, I wrote the sex chromosomes of these finches. So ZW are females and ZZ are males. So in figure A, you can see the W-linked mRNA is tagged, and it is very pronounced in the left side, which is the female side. Since females are the only ones that have the W chromosome, they're the only ones that can express W-linked mRNA. So as you can see, the right side, which is male, doesn't actually produce any of this mRNA. And then for B, since they both have the Z chromosome, but males have double the amount of expression of this Z-linked mRNA, you can see there's greater expression on the left side. And I'm describing these as like I see them, but the right and left are flipped in the figure, as you can tell. So then on the right side, this figure looks at the song centers, and they basically take the percent difference between the left and right side of the brain. So in black, you can see the gynandromorphic finch, but in gray are just controls, which are normal sex expressing finches. So as you can see, the HVC here nucleus shows like almost an 85% difference between the right and left side, which indicates that the male side had a much bigger song nucleus than the female side. And what they observed was that this finch would quite literally sing to itself. So the male side would produce this song, and then the other female side would be neurologically receptive to this song, which is super interesting. So basically the implications of this study don't correlate exactly to gender because they are, talking about a sex-linked chromosomal mutation, which can't be applied to transgender individuals. But it does signify that there is a disconnection between what the gonadal environment of an organism can be versus its neural environment. So you can have, for example, if you have a mutation, a dissonance between your gonadal sex and your neurological sex. And basically there was a review that talked about this paper, which also pointed out that aside from mutations, you can have epigenetic differences in the way that the brain receives the hormones produced by the gonads. For example, you have enzymes called aromatases, which change testosterone into estradiol. So the way your brain responds to hormones can be due to hormone secretion by the gonads, hormone sensitivity by receptors, and aromatizing enzymes. So the epigenetics of an individual can change how all of these are expressed, and therefore how it responds to hormones.
So I'm going to talk about two different papers relating to phantom genitalia and genetic associations of gender dysphoria. So the first article that we will be looking at is Phantom Penises in Transsexuals. V.S. Ramasandran was the first author of this paper. He is the director of the Center of Brain and Cognition and is a professor in psychology at University of California, San Diego. And he kind of pioneered the work on phantom limbs, which is the focus of a lot of his prior research, specifically on synaptic plasticity. So just some important terms and information to highlight. To start off, he mentions the attributes needed for a sense of gender identity and describes these attributes as a vector which interacts at a single point to create a sense of self. So this includes a sense of spatial continuity, free will, being a person, embodiment, as well as body image, self-consciousness, and self-awareness. Phantom sensations are the vivid sensation of still having a limb, even though it is not present, which kind of showed the malleability of body image and suggests that there is some innate scaffold of body image, which could be attributed to mirror neurons. So the goal of this experiment was to determine how innate one's gender-specific body image was and to find a link between gender identity and sexual body image. So a questionnaire was posted on support websites for post-op trans individuals, and they were told the main interest is phantom sensations, and the questionnaire contained many unrelated questions to avoid kind of prompting answers from participants. So there were 29 female to male participants, all of whom who had a chest reconstruction surgery, but only 9 of those 29 individuals have had genital reassignment. And an important number they used to compare the results to was the percentage of cis men that have had phantom penises post phalectomy, which is just a penis reconstruction surgery, and that's 58%. And there was also 20 male to female participants, and they were compared to cis females who experienced phantom breast post mastectomy, which is between 33 to 53%. So all of the results were found to be significant. 62% of female to male individuals experienced phantom penises in erection, and it's important to note that only 4 of the 9 individuals that experienced phantom penises that had phalloplasty experienced phantom penises, and those that did stated they experienced them before surgery. For 2 cases, phantom penises occurred briefly after starting hormone therapy, suggesting there could be a possible hormone trigger for this reaction. And then only 10% experienced phantom breast post-op, which implies the image of breast is wired in the brains of cis women, but not trans males. So for trans females, only 30% reported phantom penises post-op compared to the 58% of cis men, suggesting a difference in innate body image between males and trans females. So just some really interesting findings about the presence of phantom genitals. First, the desire to have one doesn't control its presence. So despite cultural and social experiences, these phantom penises and genitalia persist and are very vivid and detailed, and even became more salient with time. Many report that their penis had a specific length or shape different from their ideal, and phantom erections were also reported, and not only due to erotic stimuli, but at random times that were even embarrassing. So overall, this paper really showed the reality of phantom penises, which strongly supports that one's gender-specific body image and internally perceived gender have a strong innate hardwired component in the brain. The divergence between this and one's external morphology is experienced in trans individuals. So because of this disparity, there is kind of less of a perception that something is missing. And just one really interesting thing that he did mention in the paper was saying that trans individuals should not be viewed as abnormal, but as part of a gender identity spectrum, relating it to height, which brings us into the genetic component of our next paper, which furthers the idea, analyzing a genetic link between gender dysphoria and sex hormone signaling. So the first author for this paper was Eric Verlaine, who was a professor of human genetics and pediatrics at UCLA School of Medicine, and is now the chair of the Department of Genomics and Precision Medicine at George Washington University School of Medicine. So a lot of his research focused on precision medicine and sex development differences and disorders, specifically focusing on sex development and in the brain and genetics. So just again, some important information to know about the experiment itself. Verlaine looked at 12 sex signaling genes, including quite a few which have not been used in the context of gender dysphoria before. The etiology of gender dysphoria is unknown. A lot of early research focused on a more psychological component, such as family dynamics. But neuroimaging studies have showed specific regions in the brain that are more similar of trans women to the brains of cis women, and also heritability through twin studies has been noted and suggests a genetic component. And there have been other studies identifying gene associations, but a lot of them have had really small sample sizes. So this experiment included a lot larger of a sample size. So the participants included 380 trans women with a DSM diagnosis of either transsexualism per the DSM-4 or gender dysphoria per the DSM-5. And most of these participants were receiving hormone treatment. They were compared to the control group of 344 male subjects without gender dysphoria. All participants were Caucasian, and it's kind of important to note that these participants were taken from both Australia and the US, so that could affect the results, having two different cultural experiences. So DNA was extracted from either whole blood or saliva samples, and then the extracted DNA was then genotyped for repeat length polymorphisms for the 12 sex signaling genes analyzed. So there was a significant association found between gender dysphoria and ER alpha, an estrogen regulator gene that is associated with low bone density in women. So this overrepresentation could lead to a reduction in estrogen signaling. And then SRD5A2, which is a steroid reductase associated with a reduced risk of prostate cancer and could lead to lower DHT, which is a strong androgen production. Then STS, steroid sulfatus, is associated with enzyme levels, and this overpresentation is also seen in studies on ADHD, and the incidence of ADHD is 5 times more likely in trans individuals, which suggests some overlapping etiology. And SULT2A1 is associated with higher fetal sex hormone binding globulin, which is a glycoprotein that regulates circulatory sex steroid bioavailability, so it could reduce the effects of the circulating hormones. Next, 4 different allele combinations or polymorphisms were found to have a significant overrepresentation in trans individuals. So of these 4 allele combinations, 3 of them found long CAG repeats of AR, so androgen receptor, which reduces androgen signaling, and many of these polymorphisms, it may interact with other genes to affect being transgender, and the interaction between steroid 5-alpha reductase and CYP17 was formerly associated with lower DHT levels and an increase in the sex steroid precursor production. So overall, Verlaine proposes that gender dysphoria may have some polygenetic basis, where these polymorphisms increase the production of precursor steroids and testosterone to either lead to undermasculization or feminization, and these interactions develop in utero to develop sexual differentiation and can contribute to the development of gender dysphoria. It's also important to note, as I mentioned earlier, that this was at the time like one of the largest studies on gender dysphoria and is the first to use quite a few sex signaling genes in the context of gender dysphoria. Yeah, so that's everything. Thank you for listening.
And we look forward to talking to you guys about these papers in class.